ಬಾಬು ಸಿಂಗ್ ಠಾಕೂರ್ ಪ್ಯಾರಾಮೆಡಿಕಲ್ ಕಾಲೇಜಿನಿಂದ ನಾನು ವಿಜಯಲಕ್ಷ್ಮಿ ಶಿಂದೆ ತಮ್ಮೆಲ್ಲರಿಗೂ ಹೇಳಲು ಇಚ್ಛಿಸುತ್ತೇನೆ. ಫಸ್ಟ್ ಇಯರ್ ಪ್ಯಾರಾಮೆಡಿಕಲ್ ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಜನವರಿ 2020ನೇ ಕ್ವಶನ್ ಪೇಪರನ್ನು ಹೇಗೆ ಬಿಡಿಸಬೇಕೆಂದು ತಮ್ಮಲ್ಲಿ ಸವಿಸ್ತಾರವಾಗಿ ಹೇಳಲು ಇಚ್ಛಿಸುತ್ತೇನೆ. ಈಗ ನೋಡಿ, ನಿಮಗೆ 2020ನೇ ಕ್ವಶನ್ ಪೇಪರು ನಿಮಗೆ ಕಾಣಿಸ್ತಾ ಇದೆ.
1. Explain the respiration process in human being. The respiratory system is the network of organs and tissues that help you to breathe. It includes our airways. Those are nose, mouth, throat, voice box, windpipe, trachea, bronchi and bronchioles, and also lungs and blood vessels. What is respiratory system? Respiration process in human body. The lungs and respiratory systems allow us to breathe. They bring oxygen into our bodies. That is called as inspiration or inhalation, and send carbon dioxide out. That is called as expiration or exhalation. This exchange of oxygen and carbon dioxide is called as respiration. Now, types of respiration. Aerobic respiration takes place in the presence of oxygen. Anaerobic respiration takes place in the absence of oxygen. Normal respiration rate for an adult person at rest range ಅಂತಂದ್ರೆ ಮಲ್ಕೊಂಡಾಗ 12ರಿಂದ 12 to 20 breaths per minute. Okay, then respiration events. Breathing means moving the air into and out of the lungs is also called ventilation, which you are observing in the picture. Okay, external respiration. Exchange gases between the air in the lungs and the blood. This is external respiration. Gas transportation by blood. Transport of oxygen to the body cells and the return of carbon dioxide. Now, external respiration. Exchange gas between the blood and the body cells. Last event is cellular respiration. Using the oxygen in cells processes and the production of carbon dioxide. ಮತ್ತು ಇನ್ನೊಂದು ಹ್ಯೂಮನ್ ರೆಸ್ಪಿರೇಟರಿ ಸಿಸ್ಟಮದ್ದು ಒಂದು ಡಯಾಗ್ರಾಮ್ ಇದೆ. ಆ ಡಯಾಗ್ರಾಮನ್ನು ನೀವು ಡ್ರಾ ಮಾಡಬೇಕು.
ನಂಬರ್ 2, ಡ್ರಾ ದ ಲೇಬಲ್ ಡಯಾಗ್ರಾಮ್ ಆಫ್ ನೆಫ್ರಾನ್. ಪಾರ್ಟ್ಸ್ ಆಫ್ ದ ನೆಫ್ರಾನ್. ದ ನೆಫ್ರಾನ್ಸ್ ಆರ್ ಕಾಂಪೋಸ್ಡ್ ಆಫ್ ದ ಫಾಲೋಯಿಂಗ್ ಪಾರ್ಟ್ಸ್. ನಂಬರ್ 1, ಗ್ಲೂಮರಸ್, ಬೋಮನ್ ಕ್ಯಾಪ್ಸಲ್, digital converted tubule, proximal converted tubule, and connecting duct. The kidneys are important for the process of urine formation and excretion of other metabolites and form a major part of the urinary tract. Okay, these are the parts of the nephron. Now, then, which are the main parts of nephron? The nephron is divided into 2 portions, namely number 1, glomerulus, and the renal tubule, which helps in the removal of excess waste product from the body. Okay. Next, first part of the nephron is Bowman capsule. Okay, which will be reabsorption will be occurs in the Bowman capsule. ಹೆಂಗೆ reabsorption ಆಗ್ತತಿ? First part of the nephron where blood is initially filtered and proximal converted tubule folded structure connected into the Bowman capsule, which is selectively reabsorption will takes place in the Bowman capsule. Okay, then next, main functions of the nephron. The nephron uses four mechanisms to convert blood into urine: filtration, reabsorption, secretion, and excretion. These are the four main functions of nephron. Other functions is also there, that also you have to see. How your kidneys work: remove the waste product from the body, remove drugs from the body, balance the body's fluid, release the hormones that regulate the blood pressure, produce an active form of vitamin D that promotes strong, healthy bones, control the production of red blood cells. Fourth question, explain the mechanism of working of human heart. The right side of the heart receives the blood which is in low oxygen because must has been used by the brain and body. It pumps this to our lungs, where it picks up a fresh supply of oxygen. The blood then returns to the left side of the heart, ready to pump the back out of the brain and the rest of our body. The heart is a muscular organ that pumps blood continuously throughout the body. It is comprised of four chambers. Our heart is having four chambers: the right and left atrium and the right and left ventricle. The four chambers of the heart work together by alternately contracting and relaxing to pump blood throughout the heart. It supplies oxygen and nutrients to the tissues and removes carbon dioxide and also waste from the blood. It also helps to maintain the adequate blood pressure throughout the body. Heart pumps the blood throughout the body, hence play an important role in maintaining the body temperature. The human heart has a major role in pumping oxygenated blood to all parts of the body. Okay, it meanwhile contracts and relaxation rhythmically. There are two types of blood vessels associated with the heart. Those are arteries and veins. The heart is supplying oxygen and nutrients to the tissue and removing the waste from the. Question number 5. Mention the steps of recombinant DNA technology. Recombinant DNA technology means it is a method that is used to alter the DNA of an organism to obtain the desired characteristics in them. Recombinant ಅಂತಂದ್ರೆ ನಮಗಿರುವಂತಹ ಡಿ ಎನ್ ಎದೊಳಗಿನ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸ್ ಬಿಟ್ಟು ಮತ್ತು ನಮ್ಮ ಇಚ್ಛೆ ಪ್ರಕಾರ ಡಿಸೈರ್ ನಮಗೆ ಯಾವುದು ಇಚ್ಛೆ ಐತಿ ಅದನ್ನು ನಾವೇನು ಮಾಡ್ಬೇಕು, ಅದಕ್ಕೆ ರೀಕಾಂಬಿನೆಂಟ್ ಮಾಡೋದು. ದಿಸ್ ಈಸ್ ದ ಒನ್ ಆಫ್ ದ ಮೆಥಡ್. ದಿಸ್ ಕ್ಯಾನ್ ಬಿ ಅಚೀವ್ಡ್ ಬೈ ಇನ್ಸರ್ಟಿಂಗ್ ದ ಡಿಸೈರ್ ಜೀನ್. ಇದನ್ನು ನಾವು ಹೆಂಗೆ ಅಚೀವ್ಮೆಂಟ್ ಮಾಡ್ಬೋದು ಅಂತಂದ್ರೆ ಅಲ್ಲಿದು ಬೇರೆ ಕಡೆದ ಜೀನ್ ತಗೊಂಡು ನಾವು ಆರ್ಗ್ಯಾನಿಸಮ್ದೊಳಗೆ ಇನ್ಸರ್ಷೇಷನ್ ಮಾಡೋದ್ರಿಂದ. ಓಕೆ, another organisms into the genome with the help of vector. Vector is the vehicle that carries the genes of insert. The new copy of DNA that is combined by combining two or more genes
from different organism is also known as recombinant DNA. ಅಂದ್ರೆ ರೀಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಟೆಕ್ನಾಲಜಿ ಅಂತಂದ್ರೆ ಏನು, ನಮಗೆ ಯಾವ ಇಚ್ಛೆ ಈಗ ಡಿ ಎನ್ ಎದೊಳಗೆ ಎಲ್ಲ ಜೆನೆಟಿಕ್ ನಮ್ಮ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸ್ ತಂದಿದ್ದು ತಾಯಿದು ಎಲ್ಲ ಬರ್ತಿರ್ತೈತಿ. ನಮಗೆ ಇನ್ ಕೇಸ್ ಯಾವುದೋ ಬೇರೆ ಒಂದು ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ಏನೇ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ಇದ್ದಾಗ ಏನಾಗಿರ್ತೈತಿ ಅಂದ್ರೆ ಅವಾಗ ನಾವು ಏನು ಮಾಡಬೇಕು ಅಂದ್ರೆ ರೀಕಾಂಬಿನೆಂಟ್ ನಮಗೆ ಎಂಥದ್ದು ಇಚ್ಛೆ ನಮಗೆ ಎಂಥದ್ದು ಬೇಕೋ ಅಂಥದನ್ನು ನಾವು ಏನು ಮಾಡ್ಬೇಕು ಡಿ ಎನ್ ಎದೊಳಗೆ ನಮಗೆ ರೀಕಾಂಬಿನೆಂಟ್ ಮಾಡೋದಕ್ಕೆ ಏನು ಹೇಳ್ತಾರೆ ರೀಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಟೆಕ್ನಾಲಜಿ. ಈ ಡಿ ಎನ್ ಎ ಟೆಕ್ನಾಲಜಿ ಒಳಗೆ ಸ್ಟೆಪ್ಸ್ ಅದಾವ, 7 ಸ್ಟೆಪ್ಸ್ ಅದಾವ. ಅದನ್ನು ಹೆಂಗೆ ಮಾಡ್ತಾರ? ಫಸ್ಟ್ ಒನ್ ಡಿ ಎನ್ ಎ ಐಸೋಲೇಷನ್. ಡಿ ಎನ್ ಎ ಈಸ್ ಐಸೋಲೇಟೆಡ್ ಇನ್ ಇಟ್ಸ್ ಪ್ಯೂರ್ ಫಾರ್ಮ್ ಅಂತಂದ್ರೆ ಅದು ಪ್ಯೂರ್ ಆಗಿರಬೇಕು. ವಿಚ್ ಮೀನ್ಸ್ ದೇ ಆರ್ ಡಿವೋಡ್ ಆಫ್ ಅದರ್ macromolecules cutting of DNA, then restriction enzymes digestion, amplifying of DNA, joining of DNA, insertion of DNA into the host. These are the last one is recombinant cell isolation. These are the 7 steps of recombinant DNA technology. Okay, then again you can observing the process of recombinant DNA technology. Okay, what are the process? ಇದನ್ನು ಮಾಡಬೇಕು ಅಂತಂದ್ರೆ ಏನೇನು ಮಾಡ್ತಾರ? ಜೀನ್ ಕ್ಲೋನಿಂಗ್ ಆ್ಯಂಡ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಫ್ recombinant DNA, ಅದು ಫಸ್ಟ್ ಪ್ರೊಸೆಸ್. ಸೆಕೆಂಡ್ ಪ್ರೊಸೆಸ್ ಏನು ಮಾಡ್ತಾರ ಅಂತಂದ್ರೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆಫ್ ವಿಕ್ಟರ್ ಇನ್ ಟು ದ ಹಾಸ್ಟ್. ಥರ್ಡ್ ಒನ್ ಏನು ಸೆಲೆಕ್ಷನ್ ಆಫ್ ಟ್ರಾನ್ಸ್ಫರ್ಡ್ ಸೆಲ್ಸ್ ಆ್ಯಂಡ್ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆ್ಯಂಡ್ ಟ್ರಾನ್ಸ್ಲೂಷನ್ ಆಫ್ ಇನ್ಸರ್ಟೆಡ್ gene. Okay, these are the 4 steps of processing of recombinant. Sixth question, eukaryotic animal cells structure. A eukaryotic cells has a nuclear membrane. It has a mitochondria, Golgi bodies, shell wall. It also contains locomotory organs such as cilia and flagella. The nucleus has a DNA that carries all the genetic information. Prokaryotic cells has a plasma membrane, cytoplasma and prokaryotic cells. These cells has true nucleus. True nucleus means that DNA is surrounded by membrane. Has another membrane will be bonded organelles that allow for compartmentalization of functions. 7 parts of eukaryotic cells. Which are the 7 parts? ನೀವು ಡೈಗ್ರಾಮಲ್ಲಿ ನೋಡ್ತಾ ಇದ್ದೀರಿ ಯಾವ 7 ಪಾರ್ಟ್ಸ್ ಅಂಥೇಳಿ ಅಂತಂದು. No need to tell once again. Okay, what are the pictures are there? You have to draw the picture and label the diagram neatly. 7. Explain Watson and Crick model of DNA. Watson and Crick model DNA as a double helix with 2 long helical strands. Those are twisted together in their paper titled A Structure of DNA Acid. According to them, each DNA strand has direct unit known as bases. The bases along one DNA strands match the bases along the other DNA strand. A structure of DNA, Watson and Crick described DNA as a double helix that contained two long helical strands wood together. In their model, each DNA strand contained individual unit is called as bases, and the bases along one DNA strands matched the bases along the other DNA strand, which you are observing in the picture also. Then what is um, Watson crack model? Totally, that is Watson crack model is helix and two helical strands are there. They are twisted together and titled that is a structure of DNA. According to them, the each DNA is uh, discreted is known as ಬೇಸ್ಡ್ ಅಂತಂದ್ರೆ ಅವು ಎರಡೂ ಕೂಡ್ಕೊಂಡವಲ್ಲ ಅದಕ್ಕೆ ಏನು ಹೇಳ್ತೇವೆ ನಾವು ಬೇಸ್ ಅಂತ ಹೇಳ್ತೇವೆ. ದ ಬೇಸಿಸ್ ಆಫ್ ಡಿ ಎನ್ ಎ ಅಲಾಂಗ್ ವಿತ್ ಡಿ ಎನ್ ಎ ಸ್ಟ್ಯಾಂಡ್ ಅ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಡಿ ಎನ್ ಎ. ವಾಟ್ಸನ್ ಆ್ಯಂಡ್ ಕ್ರ್ಯಾಕ್ ಡಿಸ್ಕ್ರೈಬ್ಡ್ ಡಿ ಎನ್ ಎ ಆಸ್ ಅ ಡಬಲ್ ಹೆಲಿಕ್ಸ್. ಕ್ವಶನ್ ನಂಬರ್ 8. ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಹ್ಯೂಮನ್ ಐ. The eye is made up of three coats which enclose optically clear aqueous humor, lens and Victor's body. Okay, these are the three coats. The outermost coat consists of cornea and sclera. The middle coat contains the main blood vessels to the eyes and consists back forward of the choroid, the ciliary bodies and the iris. Okay, these are the structure, three main coats. Okay, now you come to the internal structure. The internal structure of the eye consists of three layers of tissue are arranged concentrically. The sclera, the cornea make up the exterior layer. The veva is the vascular layer in the middle and subdivided into the iris, ciliary body. The choroid, the retina constituents the innermost layer and is made up of nerve tissues. Now you can come to the then three coats, then in the structure of the eyes, then internal structure also three layers. Okay, which are the three layers? Functions of people: open and close in order to regular and control the amount of light. That is a people. Okay, iris control the lights level similar to the obtuer, means how much is required, that much only that will be open the eyes of the cornea and sclera. Okay, protect the outer coat cornea, a thin membrane which provides 67% of the eye's focusing power. The crystalline lens conjugate to acquire humor, then Victor's humor. Okay, these are the functions of the eyes. Okay, then now anatomical eyes: anterior chamber and front section of the eyes. Number 9. Explain the last process of human digestive. In defecation, the final step in digestion, undigested materials are removed from the body as feces. There are 5 main steps of digestion. The digestive process are ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. Some chemical digestion occurs in the mouth. The last part of the digestive tract is anus. Anus is consist of pelvic floor muscles and that are two anal sphincter muscles, internal and external sphincter muscles, which will be controlled when you are passing the stool or you are controlling the stool, that will be controlled under the sphincter muscles. They together jobs are detect rectal continents, whether they are liquid, gas, or solid, and then control when the stool should or should not be excreted from our body. Step in digestion. Undigested materials are removed from the body as feces. Question number 12. Functions of HCl. The hydrochloric acid in the gastric juice breaks down the food and the digestive enzymes split up the proteins. The acidic gastric juice also kills the bacteria. The mucus cover the stomach walls with the protective coating. Hydrochloric acid is an essential acid in the stomach. It helps destroy bacteria as well as other harmful species. It also works as a preservative of food. Okay, HCl converts the pepsinogen, which is not active, into pepsin to metabolize proteins in our stomach. Hydrochloric acid contributes to protein digestion by supplying H plus, which actives pepsinogen, the precursor to pepsin. Pepsinogen is secreted by chief cell in the gastric gland of the body. Now, gastric juice in the stomach also contains pepsinogen, hydrochloric acid, inactivate this inactive form of enzymes into the active form of pepsin. Pepsin break down the proteins into the polypeptides. Hydrochloric acid also kill the unwanted microbes present in the stomach. The mucus protect the stomach lining from the corrosive action of the acid.